Okay, so we're going to talk about the neuron, and we've got a couple of objectives here. So basically just kind of the basic anatomy of a neuron, the functions of each part of the neuron, and then we're going to talk about myelin here. So with the neuron, there are three main parts. You've got the cell body, the dendrites, and the axon. Okay, so the cell body is where the nucleus is at. It gives the structure to the cell. And really its job is to run the show, right? That's what the cell body does, especially since it has the nucleus. This is where protein synthesis occurs. Oftentimes this is where there's some integration there. Dendrites get signals and then they send it on down the axon. And so, as I just mentioned, we've got dendrites, and this is the area where neurons receive most of their information. It is possible for another neuron to send a signal, and that signal actually occurs on the cell body or even the axon. But generally speaking, the dendrites are what get information from other cells. And so if it gets the information, it sends that information onto the cell body, and then stuff happens in the cell body. Finally, we have the axon, and so the axon here takes information away from the cell body, goes towards another cell. It ends with what we call the axon terminal, so that's down here at this end. And the axon terminal is where we have the vesicles, or where the vesicles are located that store the neurotransmitter. And ultimately, the axon is what communicates with other cells. But like I said, the axon, you can have an axon synapse here with dendrites. You can actually have an axon synapse here at the cell body. And in some cases, you even have you can have an axon communicate directly with an axon. I know these are hideous drawings, so I will erase them. But generally speaking, axons communicate with dendrites, but it's not always the case. Okay, so the next thing that we've got, and you actually see it on this figure here, is we have myelin, okay? And so we've got myelin, each of these little yellow oval shapes, that's myelin, or at least that's showing myelin, okay? So here's maybe a better part. And so the question, and I know that I used this slide in 220, is how does myelination improve the speed of the signal? Okay, and if you look at it here, what's normally happening, or what's happening with myelin, is that the action potential seemingly jumps from node to node to node. Now these nodes right here, there we go. So these nodes are little tiny gaps between the myelin, which are made up of something called a Schwann cell. Okay, so myelin is just an insulator. It's made up of fat, and what it does is it prevents the movement of ions in or out. And so what essentially happens is you have an action potential start, and then you have a whole bunch of sodium inside the cell and just a little tiny bit inside. And so what happens is it comes in and it pushes the sodium forward. Now, if you recall from our core concepts, right, one of those is flow down gradients. And so we've got a high concentration of sodium and a low concentration, or a high concentration outside and a low concentration inside. Okay, and so sodium wants its space, and so it's going to go from high concentration to low concentration. What I believe also happens at the same time is you have all this sodium rushing in, and you've got a bunch of positive charges that are with each other. They don't like being around each other. Remember, positive repels positive. And so what happens is this molecule here, this sodium molecule, is pushed towards the axon, or down the axon, okay? And what that does is it in turn depolarizes this other part of the cell. Eventually, you get to this space, this node of Ranvier, and what you have in these spaces, like right here and right here, you have more voltage-gated channels. And so by depolarizing that specific part of the cell, you open a voltage-gated channel, more sodium moves in, and it does the same thing. It pushes sodium that's already inside the cell forward. Okay. Now, with an unmyelinated neuron, there's no insulation, and so what you have to do, and here's my neuron, is you have to depolarize this area, which will depolarize this area, which will depolarize this area, so on and so on and so forth, okay, until you depolarize everything, and then it releases neurotransmitter, okay? And so this is relatively slow. This happens at about 1 meter per second, whereas myelination, the myelinated neuron, a signal can travel at about 100 meters per second, All right, and so just to kind of refresh here, it leaps, it looks like this, and so you've got hundreds of areas of on the neuron that you have to depolarize, whereas with this one you go 1, 2, 3, 4, 5, 6, 7 essentially, and then you have an action potential get all the way to the end. Okay, so sodium goes in, pushes that sodium that's already in the cell forward, opens voltage-gated channels. Those voltage-gated channels are at the nodes, so they open up again, or those open up, which lets more sodium in, and that pushes the action potential forward. It's also important to know that wide diameter, large diameter neurons, so let's just say that my neuron is wide, okay, and there's a the axon terminal. The signal moves faster on that than one that's got a relatively narrow, okay, didn't draw it very well. And this, just think of this like a freeway, so more lanes, it's easier for cars to go faster, whereas fewer lanes, it's more likely to get congested. And so we can have congestion, it can slow down the signal, that sort of thing. And so that is basic overview, review of neuron and myelination.